Principles and of inheritance and variation. Principles of inheritance and variation. Very short answer questions. What is the cross between the F1 progeny and the homozygous recessive parent called? How is it useful? Cross between the F1 progeny with the homozygous recessive parent is called test cross, and it is analyzed to predict the genotype of the test organism. It is analyzed to predict the genotype of the test organism, like F1 progeny, to identify, to test whether it is homozygous or heterozygous. The cross between the F1 progeny and the homozygous recessive parent is called test cross, and it is to analyze to predict the genotype of the test organism. F1 progeny as homozygous or heterozygous. To identify that, we do the test cross. Question number 2. Do you think Mendel's law of inheritance would have been different if the characters that he chose were located on the same chromosome? Yes, because the genes for characters located on the same chromosome may show the linkage pattern. The linked genes will not show the independent assortment. So Mendel's law of inheritance would have been different if the genes for characters located on the same chromosome. So do you think Mendel's law of inheritance would have been different if the characters that he chose were located on the same chromosome? Yes, because the genes for characters located on the same chromosome may show the linkage. The linked genes will not show independent assortment. Who proposed the chromosomal theory of inheritance? Chromosomal theory of inheritance is proposed by Sutton and Boveri. Sutton and Boveri proposed the chromosomal theory of inheritance. Question number 4. Define true breeding. Mention its significance. Define true breeding and mention its significance. A true breeding line is one that have undergone continuous self-pollination, shows the stable trait, inheritance and expressions for several generations. A true breeding line is one that have undergone continuous self-pollination and shows the stable trait, stable trait inheritance and expression for several generations. Question number 5. Explain the terms phenotype and genotype. Genotype, it is the genetic makeup of an organism. Genotype is the genetic makeup of an organism. Phenotype, the physical or external appearance of a character of an organism is called phenotype. Genotype, genetic makeup of an organism. Phenotype, physical or external appearance of a character of an organism. Question number 6. What is point mutation? Give an example. What is point mutation? Give an example. Point mutation. Mutations that occur due to change in a single base pair of DNA are called point mutation. Mutation that occurs due to change in a single base pair of DNA called point mutation. Example, sickle cell anemia. Sickle cell anemia is an example for point mutation. Question number 7. What is the genotype of wrinkled phenotype of pea seeds? The genotype of wrinkled phenotype for pea seeds is smaller. It's a homozygous recessive trait. The genotype of the wrinkled phenotype of pea seed is that smaller. It's a homozygous recessive trait. What will be the phenotypic ratio in the offsprings obtained from the following crosses? Question number 8. What will be the phenotypic ratio in the offsprings obtained from the following crosses? The 4 crosses are given. Heterozygous dominant with recessive. Homozygous dominant with homozygous recessive. Then heterozygous dominant with heterozygous dominant. Heterozygous dominant with homozygous dominant. Let us see one after another. The gene capital A is dominant over gene small a. When heterozygous dominant is crossed with homozygous recessive, heterozygous dominant crossed with homozygous recessive, will get 1:1 ratio. It's a test cross. Heterozygous dominant crossed with homozygous recessive will get 1:1 ratio. Homozygous dominant with homozygous recessive. Homozygous dominant with homozygous recessive will get 100% heterozygous dominant. It's a hybrid. The progeny shows 100% heterozygous dominant. Heterozygous dominant with heterozygous dominant. We did this in F1 with F1 cross. Heterozygous dominant with heterozygous dominant will get 75% dominant, 25% recessive, or 25% homozygous dominant, 50% heterozygous dominant, and 25% homozygous recessive. That means either phenotypic ratio of 3:1 or genotypic ratio of 1:2:1 we get when we do a cross between heterozygous dominant with heterozygous dominant. Next, heterozygous dominant with homozygous dominant will get, it's a back cross actually, one of the back cross. We get 50% homozygous dominant, 50% heterozygous dominant. Phenotypically, 100% dominant will get. Next, question number 9. In Garden P, the gene capital T for tall is dominant over its allele for dwarf, small t. Give the genotypes of the parents in the following crosses. Tall and dwarf producing all tall plants, and tall producing 3 tall, 1 dwarf. When we get all tall plants, definitely the parents will be homozygous tall and homozygous dwarf. When we are getting 3 tall and 1 dwarf, 75% tall and 25% dwarf, the parents will be heterozygous tall with heterozygous tall. A cross will be a cross between the heterozygous tall with heterozygous tall will get 3:1 ratio. A cross between the homozygous tall with homozygous dwarf will get 100% heterozygous tall plants will be available. The first one is parental cross, crossing the homozygous tall with homozygous dwarf. We say it's a parental cross done by Mendel. And the second one, heterozygous tall with heterozygous tall, is a cross with F1 with F1 generation. Molecular biology, molecular basis of inheritance. Very short answer questions. Distinguish between heterochromatin and euchromatin. Which of the two is transcriptionally active? Distinguish between heterochromatin and euchromatin. Which of the two is transcriptionally active? Euchromatin. Euchromatin, the chromatin that is loosely packed and stained light is called as chromatin. The chromatin that is loosely packed and stains light is called as chromatin. Euchromatin, the chromatin that is loosely packed and stains light is called as chromatin. Heterochromatin, the chromatin that is more densely packed and stains dark is called as heterochromatin. The chromatin that is more densely packed and stains dark is called as heterochromatin. Euchromatin is transcriptionally active. Euchromatin is transcriptionally active. Question number 2. Who proved that DNA is genetic material? What is the organism they worked on? Who proved that DNA is genetic material? What is the organism they worked on? Alfred Hershey and Martha Chase, 1952. They worked with viruses that infect bacteria and bacteriophages. Alfred Hershey and Martha Chase proved the DNA as a genetic material in 1952. They worked with viruses that infect bacteria and bacteriophages. Question number 3. What is the function of DNA polymerase? DNA polymerase uses a DNA template to catalyze polymerization of the deoxynucleotides. DNA polymerase uses a DNA template to catalyze polymerization of the deoxynucleotides. It is highly efficient and catalyzes polymerization in only one direction, 5' to 3'. It is highly efficient and catalyzes polymerization in only one direction, 5' to 3'. So DNA polymerase function, it uses as a DNA template to catalyze polymerization of deoxynucleotides. It is highly efficient and catalyzes polymerization in only one direction. Question number 4. What are the components of a nucleotide? The components of nucleotide are a pentose sugar, either it may be ribose or deoxyribose. Ribose in RNA and deoxyribose in DNA. A nitrogenous base and a phosphate group. Sugar, base, phosphate. Components of nucleotide, a pentose sugar, a nitrogenous base, and a phosphate group. Question number 5. Given below is a sequence of coding strand of DNA in a transcription unit. From 5' to 3' is given here. And write the sequence of it's a complementary strand and mRNA. So complementary strand for 5' to 3' will be 3' to 5'. Should write for adenine to thymine and thymine to adenine. In case of complementary strand. In case of mRNA, for adenine, you have to replace it as uracil, and for for thymine as adenine. So the given below is a sequence of coding strand that you should remember. Complementary strand. Complementary strand will be 3' to 5'. mRNA also 3' to 5'. Only difference we have to show, instead of thymine, we need to show uracil in case of mRNA. Question number 6. Name any 3 viruses which have RNA as the genetic material. Name any 3 viruses which have RNA as a genetic material. Tobacco mosaic virus, TMV. QB bacteriophage, HIV, human immunodeficiency virus. Tobacco mosaic virus, TMV, QB bacteriophage, HIV, human immunodeficiency virus. What are the components of a transcription unit? A transcription unit contains a promoter and a structural gene and a terminator. A promoter, structural gene, terminator, collectively called transcription unit. Question number 8. What is the difference between exons and introns? Exons, the coding sequences or expressed sequences are defined as exons. In heterogeneous nuclear RNA, the coding sequences or expressed sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. Next, intron, the non-coding sequences in split gene or discontinuous genes of eukaryote that interrupt the exons. Those sequences that do not appear in mature RNA. So exons who code for amino acid, intron who do not code for amino acid. Hence we call exons as coding sequences, introns as non-coding sequences. So introns to be removed over, that these sequences do not appear in the mature RNA, they have to be removed over. What is meant by capping and tailing? Addition of methyl guanosine triphosphate at the 5' end of heterogeneous nuclear RNA is called capping. It occurs done in eukaryotes. Addition of methyl guanosine triphosphate to the 5' end of heterogeneous RNA is called capping, 5 capping. Addition of adenylate residues, adenine residues that at 3' end of heterogeneous nuclear RNA is called tailing. So 5 capping and 3 tailing. 5 capping include methyl guanosine triphosphate. 3 tailing include adenylate residues. Poly A tail we can call it as. Okay, so what is meant by capping and tailing? It's done in eukaryotes. that addition of methyl guanosine triphosphate at the 5' end is called 5-capping. Addition of adenylate residues at 3' end of heterogeneous nuclear RNA is called tailing. Question number 10. What is meant by point mutation? Give an example. Point mutation, give an example. The mutation that occurs in a single base pair of a gene is called point mutation. The mutation that occurs in a single base pair of a gene is called point mutation. Point mutation in the gene for beta globin chain in human hemoglobin, you'll find beta globin chain that results in the change of amino acid residue glutamate to valine. It results in a diseased condition called sickle cell anemia. A single base pair mutation we call point mutation. What is the result of point mutation? That in case of hemoglobin, the beta globin chain contains glutamic acid in the 6th position. Because of this point mutation, it was replaced by valine, which results into a normal biconcave shaped RBC will become sickle shape. We call it as sickle cell anemia. We say it as sickle cell anemia. Question number 11. What is meant by charging of tRNA? Amino acids are activated in the presence of ATP and linked to their cognate tRNA, which this process called charging of tRNA. Amino acids are activated during the process of translation. Amino acids are activated in the presence of ATP and linked to their cognate tRNA, which is process called charging of tRNA. We call it as charging of tRNA. Question number 12. What is the function of the codon AUG? AUG has dual functions. It codes for methionine as well as it also acts as a initiator codon. So AUG is the initiator codon and AUG is also code for methionine. Define stop codon and write the codons. The codons UAA, UAG and UGA. UAA, UAE, UGA are the stop codons. These codons provide signal to stop protein synthesis. Hence, they are called stop codons. The codons UAA, UAG, UGA are the stop codons. And these codons provide the signal to stop protein synthesis. Hence, they are called stop codons. Question number 14. What is the difference between the template strand and a coding strand in a DNA molecule? Template strand, the transcribed RNA has 5'3" polarity. It is transcribed in 5'3" direction on the 3' strand of DNA, which is called as template strand because it acts as a template for RNA synthesis. Okay, the strand which is helpful for that where mRNA that going to be synthesized over, we can say template strand. The transcribed RNA has 5'3" polarity. It is transcribed in 5'3" direction on the 3' strand. So, who is template strand? 3' strand of DNA is called as template strand. 3' strand of DNA is called as template strand. Who is coding strand? 5' strand or complementary strand of DNA is called coding strand because the sequence of nitrogenous base in the strand is similar to that of RNA. Okay, so 3' strand of DNA is called template strand. Then 5' strand of complementary strand of DNA is called coding strand. Question number 15. Write any two differences between DNA and RNA. DNA. The sugar molecule in DNA is deoxyribose. Sugar molecule in RNA is ribose. In DNA, thymine is present. In RNA, uracil is present. So the sugar molecule in DNA is deoxyribose and sugar molecule in RNA is ribose. and DNA. Thymine is present in DNA and uracil is present in RNA. Then in a typical DNA molecule, the proportion of thymine is 30% of the nitrogen bases. Find out the percentages of other nitrogenous bases. Simple here that if thymine is 30%, that their complementary adenine is going to be 30%. That means according to Chargaff rule, number of adenine equal to number of thymine. Then 30 plus 30, 60% over. So that means remaining 40% we have to give equally to the guanine and cytosine. So according to Chargaff rule, adenine equals thymine, guanine equals cytosine. In case of DNA, adenine equals thymine, guanine equals cytosine. So that's why if thymine is 30, automatically adenine is going to be 30%. Then rest is going to be with the guanine and cytosine 20%, which makes 100% total. Okay, next the question, the 17, the proportion of nucleotides in a given nucleic acid are adenine 18%, guanine 30%, cytosine 42%, and uracil 10%. Here you can see adenine is not equals uracil, and instead of thymine, uracil is there. Immediately we can recognize that is the RNA. Because in DNA thymine present and in RNA uracil present in the place of thymine. And moreover, the guanine does not equal cytosine. So by looking towards this, what we can say, it is not definitely DNA, it is going to be RNA. And it is a single strand because they are not equal. So we can say single strand. So answer for this question, the given proportion of nucleotides by using them, I recognize it as RNA, and it is a single strand because they are not equal in number. Guanine is not equaling cytosine here. Adenine is not equals the uracil. Hence, we can say it is RNA and it is single stranded.